This is Paul. Paul loves to exercise. He also loves physiology. Paul wants to know why he uses more glycogen when he exercises at higher intensities. After reading an article called Regulation of Skeletal Muscle Glycogen Phosphorylase and PDH at Varying Exercise Outputs by Howlett et al., Paul hypothesized that glycogen breakdown in response to exercise was related to the conservation of mass of ATP. Let's find out if he's right. As Paul increases his exercise intensity, there is an increase in outflow from the ATP pool. For every mass, there must be an inflow and an outflow. So how do we conserve our ATP mass? As exercise intensity increases, outflow from ATP increases, and inflow to ADP and AMP increase. So what accounts for inflow for ATP? Glycogen is a naturally occurring substrate that is stored in the liver and muscle and is used to provide energy to the body during exercise. Glycogen is broken down through physiological processes into a series of substrates that are used to produce ATP, therefore increasing inflow to our ATP pool in order to match the increased outflow. Let's take a closer look at these processes. As Paul hypothesized, an increase in exercise intensity will result in more ATP being broken down, and therefore increase inflow to the ADP-AMP pool. The accumulation of these low-energy molecules will in turn activate a number of enzymes that are responsible for turning various processes on or off. The three enzymes we'll examine to test Paul's hypothesis are phosphokinase, phosphofructokinase, or PFK, and pyruvate dehydrogenase, depicted as PDH. The first enzyme we'll examine is phosphokinase. The increase in the ADP-AMP pool activates phosphokinase, which in turn activates phosphorylase or phos. By increasing phos activity, there's an increase in breakdown of glycogen to G6P, which in turn is used to produce ATP. The second enzyme, PFK, is activated by the increase in ADP-AMP, stimulating the breakdown of G6P into pyruvate and increasing inflow for ATP. Finally, high levels of ADP-AMP inhibit PDH kinase, which in turn activates PDH. This increases inflow of substrate acetyl-CoA for the citric acid cycle, or TCA, which produces mucho ATP. A study by Howlett et al. Studied 7 healthy active subjects, 3 female and 4 male, with the mean age of 22.1 years old. Subjects were untrained but enjoyed a healthy active lifestyle. Before the experiment, subjects underwent an incremental exercise test on a cycloergometer to determine VO2 max. Subjects then practiced cycling at 35, 65, and 90% of their VO2 max on 2 separate occasions. Subjects were then tested at each percent VO2 on 3 separate occasions, with muscle biopsies taken at rest, 1 minute, and 10 minutes of exercise. Results showed that free ADP, AMP, and PI were unchanged from rest at 35%, but rose significantly at 65 and 90%, with accumulations at 90% being significantly higher than those at 65%. These results, consistent with Paul's hypothesis, suggest that the accumulation of low-energy molecules, ADP and AMP, during high-intensity exercise activate enzymes that support an increase in glycogen breakdown and utilization. Let's recap. Increasing exercise intensity results in an increase in inflow to the ADP-AMP pool. This increases the activation of phos, PDH, and PFK, supporting both an increase of glycogen breakdown and an increase in inflow to the ATP pool. Inflow matches outflow, and Paul is a happy exerciser.